Let's talk about meiosis. Meiosis is a form of eukaryotic cell division that is similar to mitosis, but it differs in a couple key ways. Meiosis results in 4 unique daughter cells, each of which has half the number of chromosomes as the parent cell. So, if the parent cell has 2 chromosomes to begin with, then there will only be 1 chromosome left in the daughter cell. Mitosis next results in 2 daughter cells with the exact number of chromosomes as the parent cell. So if there are 2 chromosomes to begin with, you will end up with 2 chromosomes in the daughter cells. This is because the resulting cells from meiosis are destined to become gametes. Gametes are an organism's reproductive cell. Well, why is this process important? First, it ensures that all organisms produced via sexual reproduction contain the correct number of chromosomes. And it also produces genetic variation or genetic diversity. Sexual reproduction even creates more diversity when the 2 gametes unite during fertilization, creating an offspring with a completely unique combination of DNA. That's why kids look like a combination of their parents. So let's start with an overview. Meiosis is often divided into 2 different sections or phases, which we call Meiosis I and Meiosis II. In each of these phases, there is prophase, metaphase, anaphase, and telophase, which should be familiar terms from mitosis. Meiosis I begins after interphase I, where proteins are grown and chromosomes are replicated. This phase is considered reductive division because the cells are being reduced from diploid to haploid. So, interphase occurs first before meiosis I. This is the resting phase. The cell grows, the chromosomes are replicated, and they prepare for division. In prophase I, condensation of chromatid, synapsis or contact between homologous chromosomes, and crossing over of genetic material between synapse chromosomes occurs. Crossing over, if you don't know, is the exchange of genes resulting in a mixture of characteristics in offspring, contributing to genetic diversity. There are also 5 subphases in prophase called leptonema, zygonema, pycnema, diplonema, and diakinesis occurring, but for this video, all you need to know about is prophase and the basic concepts. Metaphase I is next. The homologous pairs align at the metaphase plate, or the plane here, in the center of the cell. Independent assortment determines the orientation of each bivalent so that the homologous chromosomes don't end up in the same cell. Anaphase I then occurs. Microtubules shorten during this phase, and chromosomes are pulled to the opposite poles. The sister chromatids are still connected, and the cell elongates to prepare for division or cytokinesis. Finally, telophase I. Meiosis I ends when the chromosomes arrive at opposite poles of the cell. Microtubules are then disintegrated, chromatin is unraveled, and the new membranes are formed. When cytokinesis occurs, there are 2 non-identical daughter cells produced. There you go. Now that meiosis I is complete, we start the process again with the phase of meiosis II. This is known as equational division, where each haploid cell ends up with a haploid daughter cell. We start this phase with prophase II. Chromosomes condense and become visible again. No crossing over occurs this time. Then, following is metaphase II. Spindle fibers attach to each chromatid and they are aligned at the metaphase plate. Anaphase II is next. In anaphase II, sister chromatodes are pulled from each pole. Now from this point on, the chromatids are known as sister chromosomes. Finally, telophase II and cytokinesis. Chromosomes here have reached their opposing poles, spindle disintegrates, and the nuclear envelope reforms around each haploid chromosome. And that's how we end up with gametes. So here's what to remember. Meiosis I is the first phase of division. Meiosis II is the second phase. This process results in gametes for sexual reproduction and increases genetic diversity in sexually reproducing organisms. Here is another summary with different visuals. Feel free to pause and read if needed. And of course, Here are the sources if you'd like to do your own research. Thank you so much for watching and learning about meiosis in this video.